Hello, everyone, and welcome to today's webinar titled Tools for Accelerating Research Using Next Generation Cell Models. This webinar is a part of the 7th annual event in the Cell Biology Virtual Event Series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Sino Biological. For more information about Sino Biological, go to www.sinobiological.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen in the navigation bar from within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located to the right of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speaker, Shebna Massey, Associate Product Manager at Sino Biological. For a complete biography on our speaker, please find it on the left of your screen. Shebna, you may now begin your presentation.
Thank you, Sydney. Hi, everyone. Thank you for joining for this webinar today. I am Shebna Massey, an Associate Product Manager at Sino Biological. Today, I'm going to talk about the next generation cell-based models in biomedical research and what tools Sino Biological offers to accelerate the research using these next-gen cell models. I will begin with what are the different kinds of cell models and the next generation cell models that are available for research. Then I will dive a bit deeper into stem cells and organoids as the next generation cell models. And following that, I will discuss the tools that are necessary for research using stem cells and organoids. And finally, I will share about all the products and services Sino Biological is offering to support the research using stem cells and organoid models. So let's begin with the overview of next generation cell models. Animal models and two-dimensional 2D cell lines have been successfully applied in biomedical fields over the past 100 years for purposes such as improving our knowledge of cellular signaling pathways, developing guidelines for the design of candidate compounds, identifying potential drug targets, and clarifying the underlying pathological mechanisms of diseases. Scientists rely on a wide array of cells as the in vitro experimental models for research, and there are examples of primary cell cultures, immortalized cell lines, luciferase labeled cells, progenitor cells, and others. And then there are 2D and 3D advanced next generation cell models. And all these different kinds of in vitro models serve the purpose of the principles of the 3 R's of performing more humane research, where we want reduction, refinement, and replacement of animal models for research. And biopharmaceutical companies around the world are looking to reduce, refine, and eventually replace animal models for preclinical drug development and toxicology studies and disease modeling to save time, reduce the cost, and be more predictive of clinical trials. So in this, the various in vitro human cell line models have been widely used for such applications. The 2D stem cells offer their ability to self-renew, self-replicate, and differentiate into multiple cell lines. They offer a more realistic model to identify and confirm new drug targets and generate pharmacology and toxicology data earlier with stronger translation to the clinical setting. And additionally, the application of stem cells in drug development creates a new path to personalized medicine, while at the same time reducing or even potentially replacing animal testing. The embryonic stem cells, induced pluripotent stem cells, and others like mesenchymal, hematopoietic, neural stem, and cancer stem cells they provide researchers with opportunities to study targets and pathways that are more relevant to the normal and disease physiology and processes. And more recently, researchers have achieved to differentiate and direct these stem cells into 3D or cultures of organoids. And some other 3D models that are being utilized are co-culture models and organ on a chip. So the focus of this talk will be developments in embryonic and induced pluripotent stem cells models and organoid biology. So as mentioned, stem cells provide researchers with new opportunities to study targets and pathways that are more relevant to disease processes. They offer a more realistic model to identify and confirm new drug targets and generate pharmacology and toxicology data earlier. And they provide the opportunity to have a stronger translation to the clinical setting. And additionally, the application of stem cells in drug development creates a new path to personalized medicine, while at the same time, it has the potential to reduce or even replace the animal testing. Now, the embryonic stem cells are pluripotent cells with the capacity to differentiate into various lineages, of multipotent and unipotent cells, and they facilitate research models in various fields. And because of their pluripotency and unlimited capacity for self-renewal, embryonic stem cell therapies have been proposed for regenerative medicine and tissue replacement after injury or disease. On the other hand, there are induced pluripotent stem cells that they begin with somatic cells, and they are a promising alternative for studying antigen expression in vitro and developing novel cellular therapies. So these induced pluripotent stem cells, iPSCs, are derived from differentiated adult somatic cells that are reprogrammed into embryonic-like pluripotent state by using different transcription factors, such as MYC, OCT4, SOX2, and others, are capable of differentiating into any type of cell lineage. And with their unlimited capacity for self-renewal differentiation, these iPSCs hold vast potential in regenerative medicine, disease modeling, drug testing, and innovative cell gene therapies as well. So overall, the embryonic stem cells and induced pluripotent stem cells have the remarkable ability to differentiate into various cell types, as we can see here. And the process of differentiation can be directed through supplementation with specific cytokines and growth factors. So cytokines and growth factors, they play a crucial role in regulating cell behavior and development. And by adding these to the culture media, researchers can direct the differentiation of embryonic stem cells and iPSCs towards specific lineages or cell types. So I briefly mentioned that organoids are developed from stem cells. Organoids are in vitro miniaturized and simplified model systems of organs. These 3D multicellular in vitro tissue cultures that mimic their corresponding in vivo organs, they are cultures, they can be crafted to resemble much of the complexity of an organ. Therefore, they are changing the way diseases are studied and treated. Organoid technology has recently emerged as an essential tool for both fundamental and biomedical research. They are self-assembling three-dimensional structures that closely resemble the architecture and function of real organs. Organoids can be generated from pluripotent stem cells, adult stem cells, human embryonic stem cells, or primary tissues collected via biopsies. Using different growth factors and cytokines, these stem cells are directed to differentiate into different kinds of miniature organs like lung organoids, prostate organoids, mammary gland organoids, liver organoids, stomach organoids, and others. Now, let's look at how stem cells can be directed to differentiate into different tissues. Over the past decade, organoid research has developed to such an extent that organoid culture systems can model tissue structures from all three primary cell lineages. And organoids can be derived from embryonic stem cells or adult induced pluripotent stem cells or primary tissue. And different media components are required for each of these type of source cells used and the type of differentiation to be achieved. And there are growth factors that are essential components of organoid culture. So pluripotent stem cells, derived organoids are established following different differentiation of PSCs, which involves germ layer different specification for endoderm, mesoderm, or ectoderm. And it is followed by induction and maturation by culturing with specific growth factors as seen here to obtain the specific cell type and have the desired organoid, such as intestines, stomach, liver, kidney, lungs, and others. For the adult stem cells, derived organoids, they are of epithelial origin. These adult stem cells are isolated from specific tissues or organs and then cultivated in the presence of specific growth factors, as seen here, to organize and develop into distinct organoids like intestine, lung, stomach, liver, pancreas, and others. Now, here is this chart where we can see summarized information on the type, either the pluripotent stem cells or adult stem cells, and their biobanking status and their ability to model the different diseases. And from this, we can see that each type of stem cell are helpful in generating what different key organoids. And we can see whether they are biobankable and how well they are recapitulating the disease. And it is notable here that majority of the organoids are capable of modeling the disease, and they can be biobanked as well. So far we have seen what cells and factors differentiate into what organoids. Here we can see what different diseases can these various organoids be used successfully in studying. So organoids have emerged as powerful tools for disease modeling due to their ability to recapitulate the structure and function of specific organs or tissues. Like cerebral organoids can be utilized to study autism spectrum disorders, Rett syndrome, schizophrenia, and neurodegenerative diseases like Alzheimer's and Parkinson's. Optic organoids function as models to study glaucoma and other optic disorders. Then we have liver organoids that serve as models to expand research into fibrosis, hepatitis B, cystic fibrosis, and have also been used widely to study COVID-19 disease. Similarly, lung organoids are used in studying influenza, RSV, and other respiratory viruses. These are just to name a few from this very non-comprehensive list. And overall, this just presents as an example to emphasize the importance of organoids in modeling diseases. While we have emphasized on the 3 R's of research earlier in this talk and how organoids can reduce or even potentially eliminate the need for model organisms, this table here offers a side-by-side comparison of some key features between organoids and other animal models and cell culture models that are used in research commonly. So comparing the 2D cell culture in these animal models, C. elegans, Drosophila, melanogaster, zebrafish, mice models, and PDXs. When we look at all these different features, of ease of establishing system, ease of maintenance, recapitulation of developmental biology, duration of experiments, genetic manipulation, genome-wide screening, and others, we can see that human organoids are able to meet all of these requirements, and they offer suitable model to cover and recapitulate all of the features of all of these animal models and cell culture models. So they are a very suitable model to potentially replace these animal models. Now, looking into the overall biomedical applications of organoids, they have various biomedical applications, from a better understanding of development of modeling diseases to being incorporated into drug development. These organoids are being incorporated into these workflows and being used in regenerative medicine. They can be used for cell-based therapies, personalized medicine, and genome analysis. And organoid technology provides ideal models that mimic human genetic diseases caused by mutations or other diseases like we have seen various neurodegenerative diseases. And by using different gene editing techniques such as CRISPR-Cas9, researchers can investigate diseases associated with different genetic defects as well. And the organoid technology has also provided a potential tool for high-throughput drug discovery, and it enables accurate toxicity testing and preclinical studies. Advances in cancer research within organoids are opening the way for promising organoid transplantation therapy in the future. And further, in vitro organoids recapitulate the principles of organ biology, and they offer simplified and easily accessible cellular model systems that mimic specific aspects of the 3D architecture, composition of cell types, and human organ functions. So overall, organoid technology may fill a need in medical research, and it holds a very promising future for a wide range of translational applications. Organoid cultures, they have some challenges as well. So although these 3D organoid field, the 3D organoid field is moving ahead in offering very innovative approaches for biomedical research and clinical translational research, there are still several hurdles that need to be addressed. Specifically, the sizes of organoids developed to date have been limited due to the lack of vascular structures. But that can be overcome by using the vascularization via co-culture with endothelial cells. And that can provide a near physiological way to increase nutrient exchange. Then there is also a limitation of slow maturation. That is a crucial limiting factor in disease modeling of the later developmental stages. But pre-treating organoids with very small molecules, compounds, they can offer a solution to this hurdle. Then the establishment of ecto, meso, and endoderm tissue interactions during development may also be a promising approach to improve recapitulation of the typical organ system interactions observed in vivo. And also tissue engineering is also a promising approach to using microfluidic platform in composite organoids. And as mentioned, co-culture systems could offer the advantages of 3D organoid model systems to study functional and spatiotemporal interactions among diverse cell populations. So overall, emerging organoid technologies have already been useful for biomedical research, for drug screening in individualized medicine, and in combination with genome editing technology for gene therapy. And the broader applications of organoids are in the beginning stage of exploration, but with extensive research, we will be enabled to use these 3D organoids in replenishing existing model systems. And that can help us to strengthen the basic and clinical studies in the future. Next, let's discuss some key tools that are required for stem cells and organoid research. So stem cell and organoid research relies on a wide range of tools and techniques to culture, manipulate, and effectively use these cells and structures as models. Starting from various cell culture components, there are recombinant growth factors and cytokines that are essential for maintaining stem cell pluripotency and promoting differentiation when needed. Then we have the antibodies that serve in characterization. They help in detection of specific protein biomarkers in cells and organoids, and they are used in techniques ranging from IHC to flow cytometry and Western blotting. Then similarly, we have ELISA kits that are used for detection of tissue-specific markers and cytokines. And then we also have the PCR primers and cDNA clones that can be used in the screening and then genome editing and transduction of somatic cells. So we have briefly discussed that in the process of organoid generation, medium signaling are essential for facilitating cell-to-cell interaction, and these are integral to self-assembly and cellular differentiation. So cytokines and growth factors, they play important roles in transmitting information between cells and regulating immune responses and effector functions, such as promotion of cell proliferation and growth and tissue repair. So they are extensively used in basic research and in organoid maintenance, generation, and differentiation. And the common ones that we can see here from this chart are activin A, Wnt, FGS, and BMP, where Wnt, it initiates the formation of the organoid, BMP shapes the patterning, and FGF facilitates in fueling the growth of the organoid. And then other growth factors and cytokines, they help in differentiation into specific organ-specific tissues and organoids. Then to generate an organoid, typically pluripotent stem cells are cultured and subsequently grown into a fully differentiated structure with specific cocktails of growth factors required for each step, as we have seen previously. So these specific combinations of growth factors and cytokines, such as GM-CSF, activin A, BMP4, DLL4, EGF, and FGF, among others, they are essential components of differentiating stem cells, and more specifically, iPSCs into various cell lineages, as seen in this table. And each one of this special combination of growth factors, it promotes differentiation of iPSCs into macrophages, regulatory T cells, nephron progenitors, mesenchymal stem cells, lung epithelial cell lineages, hepatocytes, and others. Now I would like to take the opportunity to share with you all what Sino Biological has developed and what all we have to offer as a panel of recombinant growth factors for stem cell and organoid research, and all the other products and services that we offer for research that uses stem cell and organoids. So Sino Biological offers a comprehensive solution for stem cell research, where we have a wide range of reagents for organoid culture and characterization, starting from qPCR primers to protein, growth factors and cytokines, antibodies for characterization, gene products for genetic modifications, and ELISA kits as well for characterization. All of our products are high in purity, offer low endotoxin. They are validated for their activity, highly cited by researchers who use our products. We offer different versions of with tag and tag-free versions. They are our products are HPLC verified, And worth highlighting that we are very competitively priced, and all of our products are made in-house. So we offer a wide range of reagents for organoid culture and characterization. As we have been emphasizing throughout this presentation how important cytokines are for organoid development, Sino Biological offers more than 800 cytokine products that cover more than 250 molecules. And it covers the major families like TGF-beta, BMP, interleukins, chemokines, and other growth factors that are used in stem cell and organoid cultures. We have noted that cytokines and growth factors are very essential for cell maintenance and differentiation. And as a global leader of high quality recombinant cytokines, Sino Biological is supporting the rapid development of stem cell research through its high quality products. And here are a few examples of DLL4, GM-CSF, Activin A, FGF10 growth factors and cytokines. And you can see here that they are validated for their bioactivity in cell proliferation and different binding assays. And also these cytokines and growth factors are highly cited in reputable journals as well. So Sino Biological does not only offer high-quality cytokines, we are also very proud to offer GMP-grade cytokines for stem cell cultures, and by employing a high-quality management system that adheres to GMP standards and implementing stringent quality management and release testing criteria, Sino Biological has successfully created a range of high GMP-grade cytokines. And here are a few examples of our featured GMP-grade cytokine products for IL-2, 21, 7, and GM-CSF, where you can see that they are validated for their bioactivity in cell proliferation assays, as well as they are validated, they are tested for their purity, so they're high in purity, validated via SES page and SEC-HPLC. And also these GMP-grade cytokines have high batch-to-batch consistency. Moving on from protein products to antibody products, where cytokines are essential in culturing of stem cells and organoids, antibodies are important in characterization, like the anti-CD45, anti-CD14, e-cadherin, and CD31 antibodies. They are important in the characterization of iPSC cell differentiation into monocyte macrophages and nephron progenitors. And they have various applications of IHC, flow cytometry, and immunofluorescence. So high-quality antibodies are very critical in the characterization of cell differentiation, especially of iPSCs. I have shared that Sino Biological growth factors and cytokines are well trusted by its users, and they are highly cited in their publications. Here are two examples from the citations of our cytokines and growth factors. Here in this one on the left, our customers used our RSPO1 product for intestinal organoid generation and culturing. And then in the right, in the example on the right, we see that this customer used our EGF product in thyroid organoid culturing. And they have successfully used this in their experiments and generated this data using our products. Now, moving on, here is a non-comprehensive list of the different cytokines and growth factors that Sino offers that are used in culturing of different organoids. And the common ones that we have previously seen are Activin A, EGF, RSPO1, BMP4, HGF, FGF, among others. And this shows by organoid type which different cytokines and growth factors are used, and Sino Biological offers all of these products. This table here, I would like to draw your attention by again highlighting that our products are highly cited and they have high batch-to-batch consistency and are tested for their high purity via SEC HPLC. And specifically, I would like to highlight that our products are very competitively priced, as seen here for this specific product of the IL-18, where we have high batch-to-batch consistency. Our prices are very competitive compared to other competitors offering these products. And towards the end of our presentation, I would like to go over our ELISA kits and pair sets that we have to offer that can be used in stem cell cultures and organoid cultures. Some features of these products, these ELISA kits and pair sets, are that these are full-length proteins that are used as immunogens, and it has both the capture and detection antibodies that are monoclonal. And these kits, they are passed through stringent quality control measures, and again, they are very affordably priced. Now, I would like to end this presentation by giving an overview of Sino Biological and all it has to offer to support the research in various fields. With an extensive catalog, we offer over 6,500 proteins, 14,000 antibodies, more than 47,000 gene products, and more than 600 ELISA kits. We use different expression systems, and from recombinant proteins to monoclonal antibodies, our high-quality products undergo rigorous testing and validation to ensure the reliability and consistency. And with our extensive portfolio, we aim to provide researchers with the necessary tools to advance scientific discovery and drive innovation in their respective fields of research. And I would also like to mention that we not only have this extensive catalog collection, but we also have CRO services that are designed to support your research and development needs across various fields. And lastly, as a global company, we reach over 190 countries, and we have established a very strong presence worldwide. And we are committed to delivering high-quality products and services that meet the needs of researchers and scientists around the globe. With that, thank you all for your attention and for joining this webinar.
Thank you, Shebna Massey, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. Let's get started. Our first question is, what specific factors contribute to the controversy surrounding the use of animal models in research? And how are next generation cell models helping address these concerns?
Thank you, Sydney, for this question. So the controversy surrounding animal models arises from concerns mainly about the limited ability to be translated to humans, and there are ethical issues related to animal welfare. So there have been recent studies that have identified that biological processes that are specific to the human body, they cannot be modeled in other animals. So some of these are like brain development, metabolism, and testing of drug efficacy. So the emergence of human in vitro 3D cell culture approaches, they are using the stem cells of different kinds of organs. They have received a lot of attention because this shows a potential to overcome these limitations. And these next generation cell models, such as iPSCs and organoids that we have talked about, they offer human relevant alternatives, and they can help reduce our reliance on animal models in research.
All right, perfect. Thank you. Our next question is, how do organoids contribute to personalized medicine, and what are the challenges in implementing patient-specific organoid models for various diseases?
Uh, Organoids offer a path to personalized medicine. They allow researchers to create disease specific models from patient derived cells. Organoids, they also help to significantly contribute in personalized medicine by offering a platform for more accurate disease modeling. They also help in drug testing and tailoring personalized therapies for individual patients. So there are some challenges still that are associated with these models, and they include establishing and maintaining organoids from diverse patient samples, and truly recapitulating the disease in these organoids might be challenging because they lack that heterogeneity that is found in complex disease environment. So, of course, there are some ethical issues and cost that is also associated with personalized medicine that are all the limiting factors in this. But despite all these challenges, the potential of patient-specific organoid models in advancing personalized medicine it's significant. And researchers and healthcare professionals, they are continuing to work on overcoming these obstacles to harness the full potential of organoids in helping improve the treatments and improving the patient outcomes.
Perfect. Thank you. Our next question is, in the context of regenerative medicine, what are the key differences between embryonic stem cells and induced pluripotent stem cells, iPSCs? And how do these differences influence their potential applications?
We briefly discussed this during the presentation. Embryonic stem cells and induced pluripotent stem cells, they are both very valuable resources in regenerative medicine due to their pluripotent nature. And that means that they have the ability to differentiate into different cell types from the body. But there are differences between these two sources of cells, and they mainly govern the ethical issues that are associated with them. iPSCs, they have the advantage of being patient specific, and ethically they're less controversial compared to the embryonic stem cells. And that makes them a promising choice for regenerative medicine. And also, there is a better immune compatibility in iPSCs. There is less genetic variability, and there is also an ease of accessibility associated with the iPSCs. And that's what overall makes iPSCs a preferred choice. And in addition to that, iPSCs also have the potential to collectively advance regenerative medicine and contribute to the development of patient specific treatments because of the reduced ethical and immunological challenges associated with them.
All right, great. Thank you. It looks like we have time for one more, so we're going to go ahead and wrap up with this final question. Could you explain the significance of vascularization in 3D organoid models and how it impacts their use in disease modeling and drug testing?
Sure. We in our previous question we addressed, how important it is that organoids closely recapitulate the environment from they model the disease. So vascularization in this case is very crucial because that provides the nutrients and oxygen to the organoids, and that allows them to mimic those in vivo conditions more accurately. Better vascularization will enhance the viability and functionality of the organoid itself, and that'll make it more relevant to modeling the disease and then also for drug testing. However, there is incorporating the vascularization into 3D organoids. It's a very complex task, and it is a significant challenge. There are researchers who are working on various methods to induce and integrate blood vessels formation Some of these methods are co-culturing with the endothelial cells or using different bioengineering approaches. Ensuring proper vascularization without really overcomplicating the model or disrupting other cellular interactions, it is slightly tricky. But having the ability to replicate the vascular component of tissues, that'll really allow the researchers to study diseases and drug testing in a more realistic and clinically relevant context. And that'll help us to ultimately advance our understanding of disease mechanisms and different treatment options.
Perfect. Thank you, Shebna. Do you happen to have any final comments for our audience today?
Thank you so much for joining for this presentation, and I hope that it was helpful in learning something about the 3D organoid models and different tools that are available for it. And thank you so much to LabRoots.
All right. Thank you again, Shebna Massey, for your time today and for your important research. Before we go, I would like to thank the audience for joining us today and for their interesting questions. I would also like to thank our sponsor, Sino Biological, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 20th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.